I'm Fiona Greer, and I'd like to welcome you to our first in a series of short, informal roundtable discussions on the theme of biosimilar analytics, hosted by Biopharmaspec. Today, we are lucky to have three experts, Dr. Richard Eastman, Dr. Marius Schmidt, and Dr. Dan Mamilak, to chat about their views on how to integrate analytics, biophysics, and bioassays to accelerate biosimilar development. Richard is the Technical Director for Structural Analysis at Biopharmaspec, an independent analytical CRO with labs in the UK and US. With over 20 years' experience in glycoprotein structural characterization using mass spectrometry, he manages carbohydrate and glycoprotein analysis at the primary level and is well acquainted with the various regulatory requirements for biomolecule development. Marius is highly experienced with all aspects of biophysical analysis, previously as a senior scientist and head of AUC and AF4 at Coriolis Pharma. In 2019, he co-founded and became CEO of the Heidelberg-based Zentriforce Pharma Research, GmbH, a CRO service provider that specializes in hydrodynamic analysis techniques for both biopharma and gene therapy products. Dan is founder and president of Toronto-based contract testing lab Custom Biologics, a Health Canada audited GLP GMP compliant global service provider for functional testing and characterization of biomolecules. Dan has a background in microbiology, immunology, and protein biochemistry, and over 20 years' experience in developing and validating reliable and reproducible cell-based bioassays to measure the functional properties of biotherapeutics. Thank you all for giving up time to join us today to talk about front-loading biosimilar development with analytical characterization, integrating analytics for analysis of primary structure, higher order structure, and function. As our collective experiences of biosimilars are drawn from years of product development, the process of getting this class of drugs to market, especially in terms of testing and approval, is constantly being refined. Questions are now being asked regarding the need for extensive clinical trials, and if it is more appropriate to front-load biosimilar drug investigations with risk-based structural functional analyses. Technological advances in both hardware and software mean that detailed investigations can take place covering all aspects of primary and higher order structure and the resulting impact on functional biological activity. So the first question I want to ask the panel concerns the recent observation that certain regulatory agencies are taking a different view on the necessity for comparative clinical efficacy trials. How do you think this impacts the analytical and functional work performed as part of a biosimilar drug development? Richard, if I may start with you.
Okay, thank you, Fiona. Yes, this is a good question. It's an interesting situation that we're in now. And it seems to me that the sort of the biosimilar idea has really sort of come a full circle on itself in a way, because originally, obviously, you had the idea of comparing a biosimilar to an innovator to look at the comparative structural data from that point of view, and using this as obviously a comparative set of information about how the biosimilar compared to the innovator. But now we have a lot of sort of historical data we can look at from the biosimilars themselves. So you now have the sort of the whole wheel has turned again on itself, and there's now a larger body of data in the biosimilar world that can be drawn upon itself. So this has led certainly the MHRA, the UK regulatory health agency, which has to come up with its own guidelines now in a post-Brexit world that we live in, to look at the way it's going to evaluate things, and has taken the stance that we can use this kind of information, this greater body of information, to maybe reduce the need for the clinical data, but rely more heavily on the knowledge that having the idea of what a particular biological molecule can do based on the known structural functional properties means that you can more heavily rely upon that data and take a risk-based approach. So the absolute necessity for a clinical trial isn't there. It can be taken on sort of a case-by-case basis. Now, that's not a view that the FDA and the EMA take at the moment, but I think they're going to watch developments with interest to see how this evolves. What it does mean, of course, is that the strength of the data that you need from a structural and functional point of view is that much greater, because you're going to be placing a far heavier reliance on it than you would do in other scenarios. Now, with the technologies and techniques available today, there's a huge amount of data that can be generated from that point of view. There's a strong emphasis going to be placed on the idea of making a very solid body of data where different techniques come and support one another in terms of their structural and functional conclusions, so that what you have is a very, very solid foundation for the belief in what your molecule is capable of doing based on really robust structural functional data. And I think, bearing in mind the position that the regulatory agencies are taking at the moment, this need for strong functional and structural data has never been greater. Marius, do you have some information you could add to that, some ideas?
Yes, thank you, Richard. I think it's a quite interesting time period we currently have. So it will be quite interesting a couple of years, especially as the first authority actually considers skipping the clinical studies. The question is, however, who is going to take the risk to being the first? That's always a tricky question, and as long as the EMA and the FDA get not on board, you still have to do the clinical studies, and so I don't know if the English authority will actually skip it, as long as it's necessary for the others, because people will do it anyhow. The other question is, how could you enlarge the scientific study? So we already have quite large studies running for biosimilarity. So I think we already get a lot of data out of it. And I think there are two possibilities I see at the moment. You could extend into the new era. One would be shifting everything to a GMP environment, which I don't know if it's really necessary. And the other one would be getting a larger statistics. I don't know if this is really necessary, but there might be an option to increase the trust into the data. But it's actually also quite a good point that we have much more data on all the biosimilars as well. So it might also be interesting to compare new biosimilars to old biosimilars as well, not only to the reference product, so that we include them in future as well. So Dan, what's your opinion on the topic?
First of all, I think you both bring up excellent points. I believe the regulatory agencies want to make it less expensive to produce biosimilars so that they could reach a wider market. That was the whole point of generic drugs. 25 years ago, biosimilars are the same. With the change in thought process that the regulatory agencies in North America, Europe, and even in South America are considering, I do believe the strength and the decision-making process will lie in the strength of the structure function package. The regulatory agencies see there isn't a need. If you're making a biosimilar, do you really have to do a thorough PK study so you understand what the Cmax is? It's not relevant anymore. It's costly, and even on a humanitarian grounds, there's no need to inject these animals for data that, Marius, to your point, one can glean from other biosimilar studies of that same drug that's already been done. So moving forward, I think they will rely on a lot of the historical or empirical data. However, every biosimilar is made with a living organism, and to demonstrate that that biosimilar, that molecule, is the same as the originator, the structure function package is key. So I think there's going to be an emphasis on that. Richard, you mentioned the advancement of various analytical techniques, and I know what Biopharmaspec does, and I know, Marius, with your company as well. That is going to be key. And that's why, again, my personal belief is that the strength of structure function packages that the regulatory agencies are going to now be seeing in the future will help them realize that animal studies, large-scale pharmacokinetic dynamic studies are no longer going to be necessary.
That's a very interesting point you've made, Dan. So if I can, following on from that, then if I can ask, what do you think is the best way to develop a front-loaded characterization study? Marius, perhaps you can give some examples or case studies of how you build orthogonality and quality into your approach. That would be good.
Yeah, so especially for biosimilars, it's pretty important to run proper method qualifications upfront, and it's not always that easy to get reliable accuracy information. So accuracy is always the most difficult to assess. Everything else, linearity, precision, specificity, is quite easy to assess. That's where normally an orthogonal method comes in. So we run a lot of aggregation analytics, and in aggregation you normally have 3 different techniques. I think everyone knows the size exclusion chromatography, which is more or less the main technique used in the whole industry. There's asymmetrical flow field flow fractionation and analytical ultracentrifugation. A couple of months ago, I stumbled over a quite interesting case where we got really valuable data by applying an orthogonal method. So we had a customer coming to us, and he told me we have a monomer dimer system. So we get the result by size exclusion chromatography. This case, we measured it by AUC, and it actually figured out to be a mixture of a tetramer and a heptamer. So there was no monomer dimer in there. So it was mostly due to the fact that they used a lot of acetonitrile in the mobile phase, which actually split up the reversible oligomers. But this is not per se a different result. So we still get orthogonal information. In this case, accuracy is not that easy to align, but measuring these 2 techniques got us a better picture, because what we have seen in SEC was actually irreversible aggregates and not the reversible as well. So the question is how you compare these 2 in this case. So the second possibility to look at accuracy is running positive control samples. That's how we normally do it, and I think it's also quite often done in other techniques as well. So Dan, how do you look at accuracy and also orthogonal techniques in general?
It's a great question, and it just, even the questions, Fiona, that you're posing just highlight the breadth of this field. So Marius, I don't think there is the same need for orthogonal approaches when measuring functional testing. I believe the orthogonal approaches are on you structural people. It's critical at that stage. Why is there not a need for it at the functional testing? Because at the functional testing, you're measuring its mechanism of action. We know that it's structurally sound, otherwise it wouldn't come to us. Sometimes it does come to us and there's all sorts of messes, but typically that's the first step, to make sure that it's structurally sound. So why aren't there orthogonal methods in functional testing anymore? I believe it's due to the emergence of these very sophisticated recombinant cell lines that are used as the target cell line for these molecules. So if the molecule works the way it's proposed to, the cell line glows and you measure a glow. It's simple on paper, though, to your point about positive controls, you do have to sort of qualify the assay against a reference manufactured product. So that's really the key. So there's no point using two different reporter cell lines to give you the same example. It's not necessary. What is necessary to show, does it function like the reference manufactured product? So in every assay that we run, we always tell the client or ask them, what is your RMP? That needs to be given to us to establish the conditions so that when we run the assay and test their biosimilar, it's similar. So really that is the key. Fiona, you were asking the business about quality. Where we come in and where we've tried to sort of make an area for ourselves is running these assays under GLP. I know Marius and I know Richard, and I've had these discussions before, is GLP required and at what stage? So we're of the belief that sort of a quality system, which I know all of our companies have, is necessary for the functional testing, because I do believe ultimately one of the first things that a regulatory agency is going to look at is, you're submitting this package, what's the relative potency? What is the potency of your biosimilar relative to the originator? How does it function? And I believe that they go back from there. Then they look at the structural, then they may look at immunogenicity, et cetera, et cetera. So that's why, just to wrap before I ask Richard his comments, that's why I think that in functional testing, there's not so many orthogonal approaches to measure the mechanism of action. There were when we didn't have these fancy reporter cell lines and we had to use these primary cell lines that are difficult to grow, etc. But now it's been quite streamlined, and the hope is that this also brings down the cost of these developers. Richard, I know we've had these discussions before. Are we still aligned, or has the mood shifted?
No, I think we're still very aligned, Dan. Actually, yeah, you've made some good points about the idea of orthogonality, as has Marius with his examples there, and you're absolutely right. This is orthogonality is something that features very strongly in the way that biopharma spec approaches the types of analyses that we do, and perhaps I can just share with you, just while you were talking, I was pulling up a table that I think demonstrates this. This is taken from an article that we wrote in Bioprocess International back in April 2020, and it's just a table of various orthogonal techniques. If I can just share this with you, so hopefully you can see that.
Yes, thank you.
So really, yeah, it's just to highlight the point that as you both say, the idea of orthogonality is really important in terms of producing this self-supporting data set where you can have strong belief in the data that you generate and the conclusions that you draw from any one particular side. Now, I'm not going to go through all of the things that are on the slide here, all the various different techniques, but the idea really is obviously that if you know something about your molecule, you can start to design orthogonality in from the very beginning. You can think about the various different techniques and what they actually tell you, but also very importantly what they don't tell you. So you can't just throw the kitchen sink at this and say I've got this orthogonal data package, because you've got to be able to understand what the techniques can do and what they can't do. So for example, you might say I could use a peptide map to look at the deamidation status of a molecule and relate that back to an ICIF electropherogram, where you're looking at more acidic species in there and see if there's a relationship between deamidation and these more acidic species that you're expecting. For a glycoprotein, if you've got a complex glycosylation on there with a lot of sialic acids on, you could examine the glycan profile and compare that to the ICIF data, both before and after treatment with sialidase, to remove that. And you can see the peak profile and the collapse of a lot of these acidic species, demonstrating the acidic nature as a result of the glycosylation profiles. You could use intact mass analysis to compare various structural features of, say, a monoclonal antibody. You know, classically, you look at things like the C-terminal lysine, presence or absence of C-terminal lysine, presence or absence of pyroglutamate residues as well at the N-terminal, either light or heavy chain. And you can look for those things in the intact mass data and compare those to components that you would see in the peptide map as well. So you're able to sort of align these things. And also quite nicely with an example like that for the C-terminal lysine, that again does feed back into the imaged capillary electrophoresis data, because the lysine containing components will be more basic. So you can treat the sample with the carboxypeptidase to remove that C-terminal lysine if it's present. You'll collapse down those more basic signals. You would also have an effect of removing those components from the peptide map and collapsing down potentially the intact mass data. So you can start to design your orthogonal experiments with the knowledge of the molecule that you're working on, and use these various different biochemical techniques along with that to really give very, very solid conclusions from the experiments that you perform. And you can design these from an early stage to make a very, very concrete package with a knowledge of your molecule and how the techniques work and how you can use these techniques together to your advantage. So I'll stop sharing my screen there.
Yeah.
All right. Thank you.
Fiona, can I?
Yeah. Yes. No. Please go ahead, Dan.
I was gonna. I have a slide that I think can complement Richard's work and Marius quite nicely. Do I have a second just to?
Yeah.
To share it?
Richard, I just want to add something to what you and Marius said. First of all, the explanation of orthogonal assays, I think it's incredibly pertinent and necessary. And even Marius, when you're doing the structural characterization, it is so important to have a well-defined product when it comes for functional testing. Because when you have that, if I could share a result with you, what we and our clients like to see will look something like this. Can you see that? It's just a slide that says comparability testing. I'll try to make it a bit bigger with it. Is that still clear with everyone?
Yes.
Yeah. So essentially, this is an adalimumab Humira shown in the black curve here, and then an adalimumab biosimilar overlay. Now these are done in, this is basically a standard curve to support a comparability study that we are currently running. And what is beautiful about this is that the two molecules truly are identical, or let me rephrase that by saying they're behaving similar. We never want to say identical. And the reason we say that is because both molecules' standard curves were separately prepared for each molecule. Each point was done in true replicates, not pseudo replicates, but 3 times a standard curve was made for the purple line and 3 times a standard curve was made for the black line. And then they were run in an assay, and the fact that they overlap one another, this basically shows that whoever had done the production or the structural characterization of the adalimumab biosimilar before it came to us for functional testing did a fantastic job. So this is really what we like to see. So we don't necessarily see all the work that goes into the structural analysis before it comes to us. Marius, to your point, where we sometimes run into problems is that the buffer that the adalimumab biosimilar comes in may be different than what the reference manufactured product is in. So as a result, there could be excipients or something or another that is in the reference manufactured product that the client may not know about. And there we need a cell-based functional assay to be able to pick up subtle differences if they have an effect on the functional testing of it and the functional properties. So that was meant really just to complement the points that both of you made about the orthogonal approaches and the extensive analytical characterization that goes into it, that we're lucky that we get the clean product and that all the finicky work goes on at the front end. So
thank you, Dan, for making that point. Thank you. Now we can move on to our final question. Dan, this is something that you may be very familiar with for our final question. What is your experience of linking analytical characterization to function, i.e. an integrated strategy? What about the challenges of performing these correlations between the originator and biosimilar? And can you illustrate this with any examples?
Yeah, Fiona, I think you're touching on the holy grail of developing a package, a biosimilar package. I do believe that the structure function characterization of a molecule is the driving force that is going to bring the cost of developing biosimilars down. And a good structure function package that has the orthogonal approaches from a structural perspective, has a well-designed functional characterization of the molecule, is really what the regulatory agencies will ultimately want to see. Now, of course, each one of those facets does have its own challenges, its own criteria to demonstrate that the methods were performed with the highest quality in mind and you're generating the utmost data. But the biggest challenge really then is, from a functional perspective, is your assay also has to show that it could pick up subtle differences. Richard, you just gave a great explanation with that table showing how you would approach structural characterization to pick up subtle differences or to confirm differences or similarities. In a functional based assay where you're always testing the biosimilar to an originator, sometimes the assay does have to be sensitive enough to show that if there are structural differences, you can capture them. And an example, just to share this point, so sometimes it's easier to look at a picture, is what I'm showing here. Can everyone see that? It's 4 curves.
Yes. Yep.
So in this case, basically the 100% curve always represents the reference manufactured product. It's what you do or the state of the test molecule that really you have to be able to capture. So in various cases, if the test molecule is not as glycosylated, or if there is a subtle structural difference, or even if there's differences in the excipients, in the buffers that you have with your drug substance or drug product compared to the reference manufactured product, you want a cell-based assay or a functional assay to be able to capture it. Because that is a question that the regulatory agencies are going to ask. They're going to say, why is this different? Now, they do allow for a certain percentage difference. 15, 20% is okay. For some molecules, it's a little larger. But essentially, what you want ultimately is just that, to be able to have a cell-based assay that does pick up the differences in structural properties that could contribute to its functional properties with it. So really, ultimately, again, I go back to what I was saying. It's for that reason why I do believe that a strong structure function package is really the key, and it's going to be the driving force that us as testing labs will be able to share with developers that we work with to say, this is what you need. This is what we know the regulatory agencies are looking for. This is what will help bring the cost of developing your biosimilar down. It's my take on it as well. Marius, you've maybe not seen all this data before, but I throw it to you for your comments.
Yes. So what we are mostly doing is aggregation analytics. So we don't have that many interactions with functionality. But I had an example where we actually had a quite interesting relation to functionality. It wasn't a biosimilar, it was a generic drug, a normal generic drug. It was actually cyclosporin oil droplet distribution. And they had problems with their product. It actually performed different to the originator. And measuring the oil droplet distribution, in the end, we found out that the issue was actually in the micellar structure of their tween, and so they had a different batch of tween, which actually caused all the problems. And that's always interesting where all the difference are coming from. I know it's in this case not a biosimilar, but I think it's quite interesting story as well.
Absolutely.
Richard, how do you assess these problems?
Well, I think both you and Dan have summed it up pretty well, Marius. Really, to my way of thinking, it's you have to know your assays well. If you're going to get an understanding of the structure-function relationship, then you have to know your assays well. You have to be able to understand exactly what you're monitoring with the assay. You have to be able to tease out that information. Is it an excipient-based issue? Is it a particular aspect of the structure that you're looking at? Is it if you're looking at sort of the binding thing and it may be associated with the glycosylation? Well, you know, how does that relate to any differences that you're seeing in glycan profiles between the batches of samples that you're analysing? Is the three-dimensional shape of the molecule the most important thing? And how does that compare to some of the higher order data that you're seeing across these different batches? Is there any evidence of scrambled bridges that may be causing this? So the idea of this kind of, I think, a very, very positive collaboration between those people who look at structure and those people who look at function, and having really good lines of communication to help to bring these two very strong aspects together that are needed in order to get over this hurdle that we call biosimilarity is very, very important. You know, they're two halves that have to fuse together to tell the whole story, and to be able to bring those aspects together is critically important. And the people that do these various different testing, to be able to communicate and relate the data across those two different areas, I think is really, really important to getting this idea of a very strong front-loaded analysis.
Yeah, well put.
Great. Well, thank you to our three expert contributors for a very, very stimulating discussion. Detailed, my message, the message that I got is that detailed structural and functional investigations as part of a comparability exercise can give us great confidence in the nature of the biosimilar, providing information from which risk-based assessments can be made. And results from the orthogonal methods can provide a meaningful insight into how similar the biosimilar is to the originator, and potentially reduce the requirement for full confirmatory clinical trials. Our speakers today all agreed that structural and functional data should be viewed in tandem for full understanding in integrated studies to enable a reliable assessment of biosimilarity. So thank you for listening, and thank you to BioPharmaSpec for hosting. And please look out for our next event by visiting the BioPharmaSpec webpage or by following on LinkedIn. And thank you once again, Richard, Marius, and Dan.
Thank you.
Thank you, Fiona. Thank you. Great. Thank you very much.